Hello, Assalamu alaikum. So, here we have this history of immunology as outlined in Kube's Immunology book, which traces the development of the field from ancient observations of immunity to the modern era of molecular immunology. Here is a summary of key milestones. Ancient and early observations. In it, we see that in 430 BC, Thucydides observed the survivors of the plague in Athens did not get reinfected, suggesting immunity. It means that those people who have faced epidemic of plague then and they survived, it means they got immunity and they can nurse only the patients of plague. It means first time he gave the concept of immunity. In 1000 CE, Chinese and Indian civilizations practiced virulation. Variolation, deliberate exposure to smallpox scabs to induce immunity just by making small cuts and placing the scars of these smallpox. And then they were giving immunity to their and they are immunizing their children against smallpox. So means they are giving vaccines. So foundations of immunology in 1798, Edward Jenner pioneered vaccination by using cowpox to protect against smallpox. And in 1880s, Louis Pasteur developed vaccines for rabies, anthrax, and cholera using weakened pathogens. Weakened pathogens means attenuated pathogens which are not able to cause disease, but they can live in the body and they can provide immunity. Means they can induce immunity and they can act as vaccines. In 83, Ilimetnikov discovered phagocytosis, identifying the role of white blood cells in immunity. So it means that they have given this concept of immunity that and that is termed as cellular immunity. So Ilimetnikov got Nobel Prize for his contribution by giving this concept of phagocytosis. Then we come to this Emil von Behring, and Emil von Behring have given the concept of humoral immunity, that he immunized a sheep and then he isolated blood and then sera and then immunized another animal, and he confirmed that the immunity lies in the humoral part or liquid part of the blood, means antibodies. In modern immunology, in 1930s, discovery of blood groups by Carl Landsteiner. In 1950s, Burnett and Medawer proposed clonal selection theory explaining antibody specificity and immune memory. In 1960s, identification of T cells and B cells clarifying the roles of cellular and humoral immunity. And in 1975, hybridoma technology developed by Köhler and Milstein, leading to monoclonal antibody production. So, monoclonal antibodies are very important in the discovery process and also in the diagnosis, and also it is a proof that clonal selection theory is the right one. In 1980s and 90s, discovery of cytokines, antigen presenting cells, MHC molecules, and the development of recombinant vaccines. In 2000s present, advances in immune checkpoint inhibitors, CRISPR gene editing, CAR T cell therapy, and messenger RNA vaccines, that is COVID 19 vaccines. So, these are the major milestones in the modern immunology. So, Karl Landsteiner was an Austrian citizen and he first time discovered the blood groups, and it was a great service of humanity that he provided, and now we are able to give support to the people by blood transfusions. And hybridoma technology can be a very good source for the diagnostics and the treatment of tumors. So